Hello and welcome to the accompanying presentation for Chapter 68, the Blood-Brain Barrier. This is Amanda Zachary with the Department of Neurosurgery at The Ohio State University Wexner Medical Center. The blood-brain barrier is a dynamic gatekeeper that anatomically connects and interacts with peripheral intravascular signals, cerebrospinal fluid, and the parenchymal environment to mediate a complex system of exchange, transport, and clearance. It is a neurovascular unit that is composed of microvascular endothelium, the basement membrane or extracellular matrix, neurons, and neuroglial structures including astrocytes, pericytes, and microglia. The blood-brain barrier selectively prevents passage of substances into the brain via tight junctions or enzymatic reactions while selectively carrying small and large molecules by passive and facilitative diffusion and active transport. Endothelial cells play a key role, and specifically they differ from peripheral endothelium in three primary ways. The blood-brain barrier lacks fenestrations and is characterized by low pinocytotic activity, both of which markedly impair fluid uptake. Secondly, tight junctions are ubiquitous in brain endothelial cells and impede pericellular transport of large or hydrophobic molecules across the blood-brain barrier into the brain parenchyma. Thirdly, mitochondria are present at much higher concentration, which provides the energy needed for active transport of various proteins and factors required by the brain. Neurons play a key role in modulation of the blood-brain barrier through enzymatic reactions. They upregulate catalytic factors specific to endothelial cells and provide noradrenergic, serotonergic, cholinergic, and GABAergic innervation to astrocytes and their associated endothelial cells. Astrocytes appear to be critical in regulating blood-brain barrier permeability, albeit likely indirectly rather than through physical interaction. Blood-brain barrier polarity is additionally maintained by astrocytes, in which distribution of aquaporin 4 plays a significant role in water absorption and potentially in edema formation. Astrocytes reinforce barrier function and contact endothelial cells on the abluminal side of the blood-brain barrier endothelium. Pericytes are undifferentiated contractile connective tissue cells that localize to capillary walls, aid in regulating the growth of endothelial cells, and modulate the integrity of capillary cells. They share a common basement membrane with brain endothelial cells and act as a secondary blood-brain barrier in a manner similar to macrophages by monitoring the perivascular space and phagocytosing unwanted substances. Microglia are the resident macrophages of the CNS and provide immunosurveillance of the CNS and the perivascular space. They act as antigen-presenting cells, identifying, engulfing, and presenting foreign compounds that have bypassed the blood-brain barrier to T cells. Microglia secrete pro-inflammatory cytokines and rapidly proliferate to contain offending agents. The extracellular matrix provides physical stability to the blood-brain barrier. It is a critical anchoring site that mediates polarity at the endothelial cell-astrocyte interface. Brain endothelial cells form a key component of the anatomic blood-brain barrier, in which they form a monolayer lining the intraluminal space of brain capillaries. These endothelial cells are supported by astrocyte end feet. The low permeability that characterizes the blood-brain barrier is conferred in part by tight packing of endothelial cells and formation of tight junctions and adherent junctions. The endothelial cell layer has a luminal and abluminal membrane, which is separated by a thin layer of cytoplasm between the blood and brain. The luminal surface is in contact with blood supply, while on the abluminal surface, endothelial cells are enveloped in a basement membrane and separated into regions associated with a pericyte. Neighboring endothelial cells are connected by tight junctions and adherence junctions. The transport polarity of the endothelial cell membrane is affected by drug efflux pumps, which are differentially distributed between the luminal and abluminal membranes. Specifically, transporters such as the sodium-potassium ATPase, sodium-hydrogen, and sodium amino acid transporter are found predominantly on the abluminal membrane. In addition, shuttling proteins that contribute to transport polarity, including the glucose transporter 1 or GLUT1 transporter, is concentrated at the abluminal membrane. Endothelial cell tight junctions can also rapidly and irreversibly alter their conformation to permit plasma constituents to cross to the abluminal side of the blood-brain barrier. Depicted is a schematic of a tight junction at the blood-brain barrier. Claudins make up the backbone of the tight junction strands, form dimers, and bind homotypically to claudins on adjacent cells to produce the primary seal of the tight junction. Claudins function to selectively permit passage of cations through tight junctions. They are arranged continuously along endothelial cells, in contrast to more discontinuous localization of occludins, suggesting that claudins form the fundamental scaffold of tight junctions, while occludins secure them. Occludin functions as a dynamic regulatory protein whose presence at the blood-brain barrier is correlated with increased transendothelial electrical resistance and decreased paracellular permeability. Junctional adhesion molecules are localized at the tight junction, and in association with platelet endothelial cellular adhesion molecule 1, or PCAM, they regulate leukocyte migration across the blood-brain barrier. Membrane-associated guanylate kinase homologs are a class of proteins that form multimers that anchor tight junctions to the cell membrane. There are three main adhesion proteins, including zona occludins 1, 2, and 3. Specifically, ZO1 serves as a scaffold for tight junction formation by binding to the C-terminal of the cytoplasmic tail of occludin and the cytoskeletal protein spectrin. Depicted is a schematic of brain peel vasculature showing the permeability gradient as penetrating peel vessels become capillaries and are characterized by the blood-brain barrier phenotype. The precise vascular localization of the functional term blood-brain barrier might be extended beyond the capillary segments to CNS microvessels, as is depicted here. The permeability of the blood-brain barrier to macromolecules is determined by both tight junction-controlled paracellular permeability through cell-to-cell junctions and caveoli-mediated transcellular permeability. In addition, there is charge polarity that exists between the abluminal and luminal surfaces of endothelial cells. This polarity influences permeability of the barrier and is regulated in part by tight junctions. In addition, differential receptor expression and density on the luminal versus abluminal surfaces allow for directional transport of solutes such as glucose and ions such as sodium, potassium, and calcium. The relationship between paracellular and transcellular permeability is of crucial importance for the regulation of transendothelial permeability. This is a schematic demonstrating various transport systems that shuttle molecules across the blood-brain barrier. Tight junctions permit the diffusion of only very small amounts of water-soluble compounds through paracellular diffusion, whereas lipid-soluble agents traverse via the transcellular lipophilic pathway. Selective transport systems also exist for glucose amino acids, purine bases, nucleosides, choline, and other substances, in addition to specific receptor-mediated endocytosis for certain proteins such as insulin and transferrin. Blood-brain barrier transporters ensure delivery of critical compounds that are essential for brain function. Tight regulation of solute gradients and molecular transport is mediated by four primary transport mechanisms. First is paracellular aqueous diffusion, whereby small water molecules are able to pass through the extracellular pathway, which is regulated by tight junctions. Second, transcellular lipophilic diffusion is a transmembrane process by which substances cross the blood-brain barrier at a rate proportional to their molecular weight and lipid solubility. Next are the saturable transport mechanisms, including receptor-mediated transcytosis, wherein transport of solutes via receptor binding and subsequent endocytosis occurs. This can occur against a concentration gradient but requires energy. Example includes the transferrin receptor, which is the most widely characterized receptor-mediated transcytosis system for targeting of drugs to the brain and mediates cellular uptake of iron-bound to transferrin. The other is channel-mediated transport, which mediates influx and efflux via transport proteins. The fourth primary mechanism is absorptive transcytosis, which is an endocytotic process mediated by clathrin-coated pits and, to a lesser extent, caveolae. It is generally selective for cationic molecules and is the predominant mechanism for passage of HIV-1 into the brain. In addition to the structural integrity of the blood-brain barrier, there is an enzymatic surveillance system that metabolizes drugs and other compounds which manage to bypass the structural barrier. Three main catalytic agents regulate transportation across the blood-brain barrier: including gamma-glutamyl transpeptidase, alkaline phosphatase, and aromatic acid decarboxylase. These are concentrated in cerebral vessels. Recently, the role of cytochrome P450 or CYP enzymes at the blood-brain barrier has taken on a greater significance in local drug metabolism. Heterogeneity in the activity and expression of these enzymes may impact variability in drug response or have other pathologic implications. CYP450 enzymes are essential for the metabolism of many drugs, and while they are abundant in the liver, there are appreciable amounts also expressed in many extrahepatic tissues, including the brain. Although the total amount of CYP is less in the brain than in the liver, the localized expression of these drug-metabolizing enzymes at the blood-brain barrier and the neurovascular unit provides a potent mechanism of biotransformation in proximity to neurons. P450 enzymes catalyze the formation of neurosteroids, anticosanoids, and metabolized substrates as diverse as vitamin A and D, centrally acting drugs, anesthetics, and environmental neurotoxins. These unique characteristics allow this family of proteins to control vital functions such as neurotrophic support, neuroprotection, control of cerebral blood flow, and maintenance of brain cholesterol homeostasis. The blood-brain barrier plays a key role in many neurological diseases, as is highlighted in this chapter, including stroke, epilepsy, traumatic brain injury, Alzheimer's, dementia, Parkinson's disease, brain tumor, and infection. Current translational research that is ongoing includes studying osmotic disruption, parasympathetic stimulation, convection-enhanced drug delivery, focused ultrasound, variations in neuroimaging, and biomarkers of blood-brain barrier disruption.